Thanks for the invitation to speak to you today. The traffic was terrible getting here from across the street. But I will not talk about chromatin or transcription factors today, like Mark alluded to. Instead, I will talk to you about human RNA binding proteins. And so this is a project that we've been working on for many years. And so I'll just give you a bit of background about it. You stand with this project. So when eukaryotic genes are transcribed, they're first make what's called a pre-messenger RNA. And these pre-messenger RNAs undergo a whole series of RNA processing events to generate the mature messenger RNA. So these include capping at the 5' end, splicing, which removes these intervening sequences called introns, and it joins the exons together. There's a process called RNA editing where adenosine residues in the RNA are converted into inosine, and that changes the potential protein coding capacity of the genes. And then at the 3' end of the RNAs, they are cleaved and polyadenylated to put this poly tail on the end. These mRNAs are then exported from the nucleus to the cytoplasm, where they can undergo translation to synthesize proteins. And then these RNAs are turned over where they're degraded as well. So the important thing for my talk is that every single step that I've drawn up here is a process that can be regulated, and they're all controlled by different RNA binding proteins that bind to the RNA and facilitate these regulatory steps. And one of the steps that my lab has worked on the most is this process called alternative splicing. And so this is a process whereby if you have a gene that has multiple exons, these exons can be spliced together in different patterns. So for this hypothetical gene, you can synthesize this so that the exon, the orange exon in the middle is skipped. And so you splice these two blue exons together to give you the transcript at the top, or you can join all three of these exons together and give you the mRNA on the bottom. Both of these mRNAs will then encode different protein sequences. And so from a single gene, you can actually synthesize different proteins through the process of alternative splicing. So this serves to both increase the diversity of proteins that are expressed in a genome, and it's a very important mechanism of gene regulation. But there was a paper published from Jonathan Pritchard's lab from Stanford a few years ago, where he was doing a study to try and figure out if you take all the genetic variants that have been identified to date that cause human diseases, what processes are affected most by these mutations. And it turns out from his finding is that RNA splicing is the process that's affected by most of the genetic disorders or genetic variants that affect disease. So this plays a very key role in disease in humans. And so if you look at, this is a typical exon, intron here. These sequences I've highlighted are what we call the consensus splice site sequences. So these are the sequences that direct the splicing process. And it turns out that 9 to 10% of all genetic diseases are caused by mutations in one of these splice sites. So they affect almost every different type of genetic disease. About 25% of synonymous mutations disrupt splicing. So a synonymous mutation is a mutation where it does not change the protein that's encoded by that RNA. So it's just a change at the genomic level, which then can occur in the RNA. But because different RNA sequences can encode for the same amino acid, you can have changes that affect the RNA, but not the protein sequences. And it turns out that a lot of these types of synonymous changes affect splicing. And for some genes, it's estimated that about 50% of the point mutations that have been identified can affect splicing. So this is an example of types of ways in which mutations can affect splicing. So in this case, you have your normal gene here, but then on the bottom, you can mutate the splice site on either end of this exon, and that would cause that exon to be skipped. So that's one way. Another one here is you can have a mutation in an exon. So this is a TGT codon, which encodes for the amino acid cysteine, is changed to CGT, which then encodes the amino acid arginine. So this T to C change here is what's called a non-synonymous mutation because it changes the protein sequence encoded by that RNA. And that is something where it affects the protein, but not the RNA sequence in the sense of this exon is still spliced. But there's another case here where you would have this CCC codon, which codes for the proline amino acid. Again, a single amino acid change going to CGC. This also encodes for proline. And so if you just looked at the genome sequence, you might think this would have no effect on the gene function because it doesn't change the protein sequence. But what it does is it causes a splicing change for this particular exon. So you would actually skip 5 amino acids in the protein. But this is very difficult to tell on a computer looking at a genome sequence. And so the way that this might happen is there are sequences in exons called splicing enhancers. And so it could be that this proline codon overlaps a potential splicing enhancer sequence. So when you mutate that C to a G, you lose the enhancer sequence, so you now skip that particular exon. Or alternatively, it could be that you create a splicing silencer sequence when you make that mutation. And so now you have a protein that binds to that exon and then would cause that to be skipped. And so there's many human diseases that are caused by mutations that affect splicing. Just shown here are a few. So one of these is called spinal muscular atrophy, where this C is a T. It's a little complicated because it's a second gene actually, but this C to T change actually causes this exon to be skipped. And so you don't make as much full-length protein for that, that transcript. The dystrophin gene, again, if you have mutations in these exons, it can cause Duchenne muscular dystrophy. Again, you can have similar mutations that would cause frontotemporal dementia. And then cystic fibrosis, the severity of that disease can also be caused by different mutations that would affect the efficiency of splicing of the CFTR gene. Okay, to make matters worse, there's about 1,500 proteins, kind of similar to the transcription factors, 1,500 genes in the human genome that encode RNA binding proteins. But we don't really know what most of these proteins do, because only a few of them have actually been studied. So this is what ChatGPT might come up with for the RNA biology field. So identifying binding sites for all human RNA binding proteins, determine the functions of those binding sites, and then once we have all of that information, we can use it to predict how these mutations will affect RNA processing, biology, and human disease. And so my lab has been involved in this project. We currently call it ENCORE, which stands for the Encyclopedia of RNA Elements. This started out as a project that was part of the ENCODE project, which is the Encyclopedia of DNA Elements many years ago. But we've been continuing this as a sort of a self-sustained project. But the goal of the project is really to come up with a reference data set for all human RNA binding proteins, where for each protein, we use the periodic table as sort of a metaphor. So when the periodic table, every atom has its own, all the same properties are listed for each of the atoms. And so you can sort of compare all the atoms to one another, find functionalities between them. The goal is to do the same type of thing for RNA binding proteins, where we have the same experiments performed for every protein under the same conditions in the same cell lines using the same quality control metrics, so that we can then easily compare all of the data to one another and then come up with ideas. So we've been doing a number of different types of experiments, but the two I'd really like to highlight today are this experiment called ECLIP and another one we call knockdown RNA-seq. So ECLIP is an experiment where we cross-link the proteins to RNA in the cells, and you do this by shining ultraviolet light on the cells, and that covalently cross-links the protein to the RNA. We then use antibodies that are designed to specifically recognize the RNA binding protein of interest. We pull these down and then sequence the bound RNAs. And then by doing that, we can determine which RNAs those proteins bind to, but also where within that RNA that protein binds to. And then with a complementary set of experiments, we perform RNA sequencing of all the RNA in the cell in both control cells and in cells where we knock down the protein of interest. And we do this either using a process called RNA interference or a process called CRISPR. And so this is an experiment for the protein RBFOX2. And in control cells, you can see that this exon is included. And when we knock this protein down, we now skip that particular exon. So by combining the clip data to the knockdown RNA-seq data, we can get not only the binding data, but also the functions of these binding sites. And all the experiments that we're doing are performed in 2 different cell types. So these are K562 and HepG2. The K562 cells are a leukemia cell line derived from a 53-year-old female, and HepG2 cells are a liver cell line derived from a 15-year-old male. So this allows us to compare the data between 2 cell types, but also between different tissue origins and also between male and female cells. And so the first step that we actually had to do was identify all these antibodies that recognize the RNA binding proteins. And we did this by literally scouring the earth for antibodies. And this is a slide from many years ago, but we basically have acquired thousands of different antibodies and tested all of them for which ones work and which ones don't. And so this is just one example for this protein TIA1, where we show that this protein can be pulled down efficiently by that antibody. And then the experiment on the right is showing that when we knock down that protein, the band that is recognized by that antibody disappears, suggesting that we pull down a protein of the right size. And then this experiment on the right is showing that it's probably the right protein because it disappears in a sequence-specific manner, because we're using sequence-specific reagents to validate that. And so, as I said, we've tested, acquired over 2,000 antibodies, and we have validated about 1,100 of these to human RNA binding proteins. And we published a paper a few years ago for sort of a freeze in the data set. At that point, we had over 1,000 different replicated data sets that have been generated. So, and we had at least one experiment performed for 356 RNA binding proteins. And in this slide, all the proteins are shown in rows, and all the different experiments or features for those proteins are shown in the columns. And so this sort of bluish purple section here is the functional annotations of these proteins. So these proteins are involved in a broad spectrum of different activities, from splicing to translation to RNA localization. But about 25% of the proteins that we've characterized have no known function in biology. They just are annotated as an RNA binding protein, or they've been identified in an assay to characterize RNA binding proteins. So this provides us an opportunity to provide functionality to a lot of different proteins. And so this is one way we would then intersect these data together. So this is a case where we would have the PTBP3 gene, and we're looking at how the protein, which is a related protein called PTBP1, regulates the PTBP3 gene. It's a lot of names. But anyway, if we're looking at this particular exon here, and if we blow this up from the clip data, which again is looking at the binding of that protein in vivo, we see nice peaks for that protein binding right next to this regulated alternative exon. And in the knockout experiments we see in control cells, that exon is not included at all. But when we knock down the protein, we now see inclusion of this exon here, which suggests that the PTBP1 protein binds to this region and represses splicing of the exon here. So that's how we can sort of integrate these 2 data types together. And then this is just a figure depicting different types of binding profiles for some of the proteins. So the PRPF8 protein here is involved in splicing. It binds to 5' splice sites. The U2AF2 gene is also involved in splicing. It binds 3' splice sites. RBFOX binds to intron sequences. IGF2BP1 binds to UTRs. And the FXR1 gene binds to coding sequences. And then we can characterize all these proteins. So again, the proteins are shown on the x-axis. And all of these proteins bind to coding sequences. The ones over here in blue bind to intron sequences, and these over in orange bind to non-coding sequences. And so for the proteins that we don't know what they do, this type of analysis can provide some information on what they might be doing. And then we can look at individual RNAs and identify proteins that bind to them. So this is a gene called exist, which is involved in dosage compensation. And we've identified 4 different proteins that bind to this transcript, but they all bind it in different regions of the RNA. So it's not like they're all binding to exactly the same regions on the RNA. They sort of bind in different patterns on that RNA. And then finally, for the data slides showing how we can use this information to start predicting how genetic variants might impact disease. This is a case for this UTRN1 gene. There's a G to C change that's been identified in a patient with a genetic disorder. And it turns out that that mutation overlaps the binding site for this protein RBFOX1. And if you look at the knockdown RNA-seq data in control cells, this adjacent exon is included. And in the knockdown cells, it's skipped. So our prediction is that this RBFOX protein binds to this region of the RNA and represses splicing of this particular, activates splicing of this particular RNA. So when it's knocked out, you now lose expression of the protein. And so this is just an update. So we now have over 1,500 replicated RNA-seq data sets that have been generated. And we have at least one data set for 762 RNA binding proteins. So we have still a way to go, but we're making progress. And then the last few slides are just showing like how you can access this data. So this project is part of a community resource project that started as part of the ENCODE project. So you can get a lot of the data there, but because the ENCODE project is ending, we're continuing this on our own. So we have our own database that we've set up, the ENCORE database. And this is designed to be more user-friendly, I would say, than the ENCODE data set. So rather than just being able to download raw data, you can get processed data. So you can look up any protein that you want to, and you can get lots of different information on this. For example, this one is showing where in the RNA or where in the transcriptome this RBFOX2 protein binds. So it gives you all the different transcripts that it binds to, but the genomic coordinates of all the different binding sites. You can also look up your favorite RNA of interest, and it will give you all the different proteins that binds to that RNA and where within that RNA that protein binds. And so we hope with this project that we're going to be able to generate a large data set that's a resource for the community that you can then use to generate hypotheses about how various aspects of RNA biology is regulated, and also understand how different genetic variants might impact human disease and how the RNA processing plays a role in that. And so I'd like to thank people in my lab who've been involved in this project. This is a collaboration for many years with Jean Yo's lab. And earlier on in the project, we also collaborated with Chris Burge and Eric Lukoye. And I have to thank NHGRI funding, and we've had various program officers throughout the years, but our current one is Stephanie, who's here as well. So thanks for your attention. Be happy to take any questions.
How is RNA splicing affected by the overall architecture of the genome, be it by expressing different RNA binding proteins or by affecting the splicing as it happens?
Yeah, that's a good question. So there's a huge amount of work going on right now studying co-transcriptional splicing. So most splicing happens co-transcriptional. So while the polymerase is still cruising along the RNA, and there's definitely feedback from chromatin to the RNA processing machinery. It's still somewhat mysterious exactly mechanistically how that actually works. But phenomenologically, it's very clear that there's certain chromatin marks that are associated with alternative exons versus other exons. But exactly how the whole processing works and the interconnection between them is still a bit mysterious.
Do you observe cell type specific RBP binding events? And what's the rough percentage of these events compared to non-specific binding events?
Oh, that's a very good question. Yes, so we do observe cell-specific binding, but it's really restricted to the genes that are expressed in one cell and not the other. So if the same gene is expressed in both cell lines, the same protein typically binds it to the same location in both cell lines. So the vast majority of cell-specific binding is really restricted to genes that are expressed in one cell and not the other.